Okay, so hi everyone. Thank you for being here for my PhD defense. I'm very happy to present this work I led for 5 years now at the Paris Brain Institute, for which I was supervised by Nathalie and Camille Jeunet, which are here. And this work is about beta regulation with EEG neurofeedback and its mechanisms and application in Parkinson's disease. So first, what is Parkinson's disease? It is the second most prevalent neurodegenerative disorder in the world and the fastest growing one, with about 2 new diagnoses every minute. So this disorder is caused by the degeneration of neurons inside of the basal ganglia, and specifically the substantia nigra neurons, as you can see on the left, by the discoloration of the structure here compared to the healthy control on the right. So this degeneration causes a wide range of symptoms, and most famously motor symptoms, among which you can have rigidity, tremor of the hands, as well as bradykinesia. So I will now illustrate bradykinesia for you. So during finger tapping, this is bradykinesia, the involuntary reduction in the amplitude of the tapping movements of the patients. Along the progress of the disorder, you can have gait disturbances appear, here shown by the loss of the heel strike and the reduced arm swing, as well as reduction in the amplitude of the steps of the patients, which is called shuffling gait. So to alleviate these symptoms, there are 2 main lines of treatment. First, dopamine replacement therapy, and second, something called deep brain stimulation. So deep brain stimulation, as you can see on the left, is about implanting an electrode inside of the brain and sending an electrical current to alleviate the symptoms. This method allows researchers to record the activity in a structure that is called the subthalamic nucleus. By processing this electrical activity, researchers have identified a pathological activity in the brain of the patients that is called beta activity. So this activity is in the 13 to the 35 Hertz band. And what they found is that beta is directly associated with the degeneration of the neurons in the patients. However, beta activity had long been described prior, and it had actually been identified in the sensory motor cortex of healthy participants. So this activity is quite famous, and what we know about beta is that it's key to normal motor functioning. So on the left, you see the historical recording of beta activity in someone that is in a relaxed state. And the famous result is that when you make a movement, for example, clenching your fist, this silences the beta activity, as you can see here. So I will refer to this reduction as a desynchronization during this presentation. What about the patients? In the patients, in the subthalamic nucleus, what we know is that beta activity is linked to the motor symptoms. The more severe the motor symptoms of the patients, the more beta activity is recorded in their subthalamic nucleus. Additionally, when the patients are given their treatments, this beta activity is reduced. And critically, the amount of motor improvement of the patients correlates with the amount of reduction of the beta activity. Finally, researchers have recently identified that the beta activity from the cortex is coupled with the beta activity from the subthalamic nucleus. So this has led the brilliant researchers of our team to come up with the following objective. Can we train the patients to modulate beta activity non-invasively, meaning in the cortex, and voluntarily to reduce their motor symptoms? So how can we do that? We are going to use a method that is called neurofeedback. So for those of you who saw the talk of Maryam this morning, she introduced a lot of the concepts, but I will go over them briefly. So neurofeedback is about training voluntary control over some neural activity. So here we record the activity of the brain using something called electroencephalography, so EEG, and we are going to process this activity in real time to extract the neural activity that we want to train. The key element is to close the loop by providing real-time feedback on this activity to the participant. So this feedback allows the participants to be aware of the success of the modulation of their neural activity. In our case, we are going to extract the beta activity, as you can see on the left, And we are going to ask the participants to imagine that they are doing movements. We do motor imagery because we know that in a similar extent to when you actually do movements, when you imagine a movement, this reduces your beta activity. So this sounds like a good idea. Why isn't neurofeedback already available for patients? You have two main challenges that haven't been solved yet. First, there are very high failure rates in neurofeedback. So we are talking about 30 to 50% of healthy young participants not managing to control the brain-computer interfaces. On top of this, the mechanisms of neurofeedback remain unclear. So we have not yet settled the effects of the experimental, psychological, and neurophysiological factors. So to tackle these challenges in my PhD, first, we focused on exploring the factors that may improve the success of neurofeedback. We did so in 23 healthy participants first. We focused specifically on whether providing transparent feedback, I will go back on this concept in the following slide, can improve the performance, and whether this is linked to the subjective sense of control of the patients and the participants. Additionally, we explored what were the neural markers of performance. In a second time, we wanted to see whether we could translate these principles to the patients. We did so in 15 patients, and we assessed whether patients can modulate their beta activity using neurofeedback and whether this is associated with motor outcomes. So for our first study, as I said, we wanted to see what factors could improve the success of neurofeedback. So this is the closed loop I was telling you about. First, to really understand the concepts, I want you to imagine that you are doing the neurofeedback protocol. So you are being asked to imagine a right hand closing by feeling the sensation in your hand, and you are being asked to focus on this blue bar on the screen. So the higher the blue bar, the more success you have in your imagery. This is quite a difficult task, actually, and this has led researchers to think of this feedback as a main effect in the failure of neurofeedback protocols. To respond to this issue, quite a few improved feedbacks have been proposed in the form of virtual reality, robotic hands, as well as multimodal feedbacks with tactile and visual components. When they assessed the effects of these feedbacks, researchers have found improved performance. So these work for success. However, we do not yet know precisely why these feedbacks improved the performance. In this experiment, we focused on a key psychological concept called sense of agency. So agency is the experience of oneself as the author of one's actions. If I take a simple example, let's say I blow on a leaf, I would expect it to fly away. And if it actually does, the matching between my expectation and the result of my action allows me to understand that I caused the outcome. This is called internal causal attribution. Can you see the parallel with neurofeedback? In neurofeedback, you're being asked to imagine a movement, but what you see on the screen is this blue bar going up and down. What we hypothesized was that when showing a virtual hand feedback that would match the movements imagined by the participant, so providing transparent feedback, this would improve the sense of agency of the participants and maybe the performance of the protocols. So we wondered whether feedback transparency can improve the performance and if this would be mediated by a stronger sense of agency. So to test this, we recruited 23 participants and we had them train with 3 different feedbacks. First, on the left, the abstract feedback in the form of a pendulum going up and down, going left to right. Second, we introduced the virtual hand that would repeatedly open and close following the motor imagery of the participants. And finally, we tested a multimodal condition where, addition to the visual hand movements, you would have intermittent vibrations on the top of the hand of the participant to elicit motor illusions. So this is the closed loop that we implemented. So we filtered the activity over the left motor cortex in the 8 to 30 Hertz band, and we filtered this activity in real time. So this is the beta activity, and you can see when it drops below a certain threshold, This makes the virtual hand on the right close. So the objective for the participants was to close the virtual hand as much as possible by doing motor imagery. So what did we find? What I'm going to show you, this metric is called neurofeedback performance. This quantifies the amount of reduction compared to the resting state level. So a 0 means no change in the beta activity, and a positive number is a successful reduction of beta. And these are the 3 feedback conditions on the bottom. So first, we found that there was better modulation of beta activity with the visual hand than the visual pendulum. This was expected based on our hypothesis. However, when we went to look at the multimodal condition, we actually found that there was a reduction in the performance of the participants. We were also interested in agency. So we assessed the level of agency of the participants by asking them whether they felt like they were the one controlling the movements of the hand or the pendulum. And this is what we found, a quite parallel result. So the best level of agency was obtained with the visual hand than the other 2 conditions. So we wondered about the relationship between the 3, feedback, agency, and performance. And when we tested these effects, we found that there was a significant link between performance and agency, in the sense that participants with the highest levels of agency were the ones who got the best performances. And critically, this relationship did not depend on feedback. So once we settled this question, we, in a second time, wanted to see what would be the neural markers of performance. So why is this important? Let's take a step back and think of our objective. What we want to do is provide someone with instructions and feedback, and we want to end up with positive motor effects. What's supposed to be in the middle is a selective beta modulation. We only want to modulate this beta activity to cause the motor effects. However, what researchers have found is that sometimes other frequency bands, such as the theta or the alpha band, can also be modulated. Additionally, sometimes you see modulations in non-motor brain regions, such as the cerebellum. So why could that be? When you are doing neurofeedback, you're not just doing neurofeedback. You're also trying to focus your attention on the task, and you are learning in the process. Additionally, you receive feedback continuously, for instance, tactile feedback, and each of these different aspects can have their own neurophysiological correlates that may not be in the beta band. So we looked at our data to test what would be the neural markers of performance beyond the targeted beta activity, and whether this would interact with the psychological and experimental factors in our study. To do so, we looked at this relationship between neural activity on the left and performance on the right. We explored neural activity in the space of the 28 electrodes of our EEG setup and the 3 to 30 Hertz frequencies, so theta, alpha, and beta frequency bands. And we extracted this relationship, which can be interpreted as such. In red, the positive slopes means more neural activity predicts better performance. And in blue, reduced activity predicts better performance. So this is what I'm going to present to you here, not the neural activity, but the relationship between the activity and the performance. When we looked at the beta band, what we found was that reduced activity over the left motor cortex predicted better performance. So beta was the band that we targeted during neurofeedback training. So this was a quite expected result. However, when we went beyond this frequency band in the theta band, this is what we found. We found that theta activity in the frontal and central areas of the brain predicted better neurofeedback performance. So we wanted to see why there was this positive relationship between the two when we did not train participants to affect this theta band. We looked at the feedback component, and this was not at play. But when we looked at sense of agency, this is what we found. There was a significant interaction between agency and theta, which can be interpreted as such. In color, you have the sense of agency. So you can see that in red, when someone has a very low sense of agency, This is when their performance depends the most, as you can see here by the red line, on theta activity. In contrast, in green, the participants with the highest levels of agency are the ones whose performance depends the least on theta activity. So to recap this first study, we found that visual transparency seemed to improve the performance, and this seems linked to the sense of agency. However, multimodal feedback unexpectedly did not improve the performance. When we analyzed the data, what we found was that sometimes the tactile component, meaning the vibrations, were not matching the visual component on the screen. So you could have movements, but not vibrations, and the opposite. So this incongruence is known to severely decrease sense of agency. So this may be the effect that would cause the decreased performance. Additionally, we found that theta supported performance, especially in participants with low senses of agency. This theta in the frontal central areas is very famous in neurofeedback for being a correlate of either mental effort or conflict processing. So you could interpret this as saying that participants with low senses of agency may have engaged more mental effort to manage successful performance in the task. So to conclude, putting effort into designing feedback that fosters or at least preserves the sense of agency may be not only key to improve the performance, but also to improve the specificity of the neurophysiological modulation. Because as we saw, participants with high sense of agency did not seem to modulate their theta activity. In a second time, we wanted to translate these principles in the patients. Just a bit of a history about neurofeedback in patients. There had already been attempts of neurofeedback with patients since 2002. However, the researchers have mostly targeted other frequency bands, such as the theta band and the occipital alpha band. And when you look at their results, they found quite weak and inconsistent motor outcome. And since then, a new field has emerged, which is called subthalamic neurofeedback. So in this field, you have an invasive recording of the activity in the subthalamic nucleus, and by directly training patients to reduce it, the researchers have found quite promising results. In our experiment, we try to bridge the gap between these two fields by testing whether providing cortical beta neurofeedback with the virtual hand feedback could be associated with positive motor outcomes. This is what we focused on. Can the patients achieve beta downregulation with neurofeedback? And is this associated with motor performance? To do so, as I said, we recruited 15 patients. So these patients were deep brain stimulation candidates to be operated in our team. These patients had quite severe motor impairments that was highly dopaminergic sensitive. In this experiment, we contrasted two conditions. On the left, in red, the patients were only imagining the movement, but they did not receive any feedback on the screen. And on the right, in blue, they received additionally to their motor imagery the visual hand that we tested in the first experiment. So we wanted to see the motor outcomes of this training. To do so, we used a clinical task called finger tapping, which you saw at the beginning. So the objective for the patients is to make as fast and as ample movements possible during 7 seconds. So what we did was follow each trial by the 7-second tapping task. The patients performed 3 to 5 sequences of this training depending on their fatigue levels. So these are the results. This is still the neurofeedback performance metric, but on the left, this was computing in an offline manner to reflect the beta reduction. What we found was that there was a better regulation of beta activity with the neurofeedback than only the motor imagery task. When we looked at the neurophysiological pattern, here on the C3 electrode in the targeted band, so these are time frequency maps. You have the 25 seconds of the trial here and the frequencies in ordinates. When you look on the right at the neurophysiological pattern during neurofeedback, you can see in blue the strong desynchronization that was quite prolonged during neurofeedback. But you can notice that it extended below into the mu band. In contrast, in motor imagery only, you have a quite weak desynchronization. When you look across the whole scalp of the patient, you can see that this desynchronization is quite bilateral in the neurofeedback, and you see the same weak pattern over the whole scalp for the motor imagery only condition. We wanted to focus more about the neurophysiological specificity of our results, so we looked beyond the beta band into the mu band. Sorry, so this was significant. We looked into the mu band, and this is what we found. There was a very strong desynchronization over the left and right motor cortices that was not present in the other condition. And it was rather replaced with a synchronization over occipital areas. And this was strongly significant. So these were for the neurophysiological results. What about the motor effects? This is tapping frequency. This means the number of taps that patients were able to do during the 7 seconds. What we found was faster tapping frequency following the neurofeedback trials than following the motor imagery only trials. Finally, we wanted to see whether the link with agency was present in the patients. And we did not find any significant relationship between agency and neurofeedback performance in the patients. To recap, we found that neurofeedback is feasible in quite a big population of patients and actually improves the modulation. There were faster tapping levels following neurofeedback trials, but the neurophysiological effects were not fully selective with a spillover into the mu band. How can we interpret this? One way of interpreting it may be that there could be a strong coupling between the mu and the beta band in the patients so that they cannot only modulate one band. Another thing is that mu is quite known for sensory-motor integration. So having this strong matching between the motor imagery, the action observation with the closed loop may have mobilized a strong sense of sensory-motor integration in the patients. Finally, we saw no link between neurofeedback and performance and agency in the patients. Why is that? A hypothesis could be that patients have a reduced sense of agency. So this is how agency can be modeled. You have the intent to move, and you predict the sensory result of your action. If this matches the actually perceived feedback after your action, this elicits sense of agency. However, in the patients, you have quite a few situations where this rule is broken. For instance, think of tremor. You have no intent to move, yet you have a movement, so a sensory feedback. In freezing of gait, the symptom where someone wants to walk but is suddenly frozen in place, you have the intent to move, but you do not have any movement. So we may think that patients have a disturbed sense of agency. So we went to look at the levels of agency of the patients compared to the healthy controls. And we did not find any significant difference between the two. So this disturbed agency may not extend in virtual situations. Additionally, this may be a dopaminergic effect. So the patients were on their dopaminergic treatment during the training, and dopamine is known to boost a component of agency called action binding. So there may be a ceiling effect where the actual performance of the patients is decoupled from their actual performance and their sense of agency. Finally, this may be interpreted as a cognitive effect. Most notably, we tested our feedback in healthy controls. But for the patients which are older and less experienced with technology, the feedback may be more difficult to interpret. So I will start the discussion by recapping the key findings. We found that there was better performance and agency with the virtual hand than the pendulum, and a significant link between agency and performance. Additionally, there was a strong link between theta and performance, especially with low senses of agency. In the patients, there was better beta downregulation with neurofeedback, no significant relationship between agency and performance, and faster tapping frequency following neurofeedback. So I think an important thing to consider for the future is further refining our protocols. So this is the closed loop, and I think that both sides of the closed loop need to be looked at. On the right, you have the experimental and psychological aspects. We saw that in the patients, agency did not seem critical to succeed in the task. So what other processes could we leverage? And one element could be providing choice to the patients. So this is an idea from Brian Lowe, which is here in the assembly. What we found is that in a 2015 study, where they providing choice to athletes doing golf putting, when they were allowed to choose the color of their golf ball, this improved their motor performance. And similar effects have been found in motor learning in patients. So you could extend this by giving choice to the patient over a ball that could be placed in the hand on the screen. Another key lead, as Maryam presented this morning, could be gamifying the training. So on the right, you can see the screenshot of a game that was implemented for patients in the team. So this game trains the patients to perform their rehabilitation movements in an engaging way. And this game was found to boost the commitment of the patients to the training. In the brain-computer interface field, gamification is also known to increase the performance. So this could look like goal setting for the patients, social collaboration, as well as real-time adaptation of the difficulty. Finally, we should look into improving the neurophysiological aspects of the loop. First, I think one very important element is to individualize the target. So what I'm going to show you is the individual neurophysiological effects in one patient. In black, you have the resting state level, in red, the neurofeedback, and in green, the motor imagery only. These were two patients where the modulation was quite expected. You have a reduction in the targeted beta band, and as we saw, in the mu band. However, in other patients, you have surprising results, such as this patient where there was an increase during neurofeedback in the mu band, and another one when this happened during motor imagery only. So one way to take this diversity into account could be to use this recent method called spectral parameterization, which allows to identify the individual peak at rest of the patients and to use this for training. Another important element is to rethink the way we consider beta activity. So this is something from a review article that we are currently preparing. And what we did was identify that there are actually two subcomponents of beta. In the low frequency band, low beta is primarily located over the primary motor cortex, so M1. And on the right, the high beta activity is primarily on the sensory motor area, so the SMA. And this is not just about the cortex. You also have different couplings with the subthalamic nucleus, which reflects very important pathways in the patients. And this has critical importance because as you go back to the symptoms of the patients, you can actually see different associations. For instance, rigidity seems primarily associated with high beta activity in the cortex. And when you look at bradykinesia, there seems to be a predominance of the low beta activity association. So we started to look into this in healthy participants. This is the work of our Master 2 student, now engineer Lily Pereira. What we did was look into whether low and high beta have different roles. We did so using neurofeedback and testing for different links with motor behavior. What I'm going to present to you is the associations between reaction times, so the speed in movement initiation, and either the reduction in low beta activity in the primarily motor cortex or high beta in the sensory motor area. And briefly, we found that there was only a significant relationship with the low beta activity, but not the high beta activity. So this is quite reminiscent of what we saw before, because as we saw, bradykinesia seems associated with this low beta component. And bradykinesia is defined partly by the reduction in speed. To my understanding, this has great promise for neurofeedback with patients, where we could aim for identifying the specific symptom signature, such as here, the physiological pattern during freezing of gait, and follow this by neurophysiological training over this pattern in order to make patients converge toward a normal neurophysiological pattern. And this would be with the ultimate goal in line to have a real-life transfer where the patient during a symptom can reuse the strategy that they learned during neurofeedback to abolish their pathological neural activity and hopefully alleviate their symptoms. So thank you for your attention. I would just like to thank deeply my two supervisors, Nathalie and Camille, as well as my direct collaborators, which I will now quote, Brian, Laurent, Léa, Emeline, Cassandra, and Lily, which are all here. And finally, the team, which I had the pleasure to interact with now for 5 years. Thank you.
Thank you very much, Claire.
Est-ce qu'on peut remettre à l'image Raphaëlle Roy et David Devos, s'il vous plaît? Alors peut-être il faut que tu... Je ne sais pas s'il faut que tu quittes le plein écran. Oui, Raphaëlle, c'est bon, on vous voit et c'est à toi.
Parfait. Thank you very much, Claire. So now we'll move to the questions from the jury. So if that's okay with the jury members, we decided that before the defense. We will start by 2 questions each for a first round of questions, and we will start with the reviewers. So first, Maryam, if you want to start.
Yeah, thank you, Raphaëlle. So, hi, Claire. Thank you very much for this very nice presentation. Before I ask my question, I would like to applaud your work in this line of research. Very interesting studies. I can imagine it's really difficult to secure Parkinson's disease patients and work together with them. So thank you so much for the contributions. Obviously, I do have questions, which I also included some of them in my report. I'm going to start with study 1. Okay. The one that you did with healthy patients, and I have 2 questions. The first question is that in your report, in your manuscript, you define the conditions that you devised in your experiment as feedback transparency. And I found that very interesting terminology, because on one hand, you had no agency condition, and then in the other hand, you had visual feedback that improved agency, and there was the multimodal feedback. So my first question is more about a clarification on why you coined this terminology.
So first, this terminology originates from the HCI field, so the human-computer interaction field. And this actually describes the quality of the mapping between the intent when you operate with technology and the actual feedback that you get, not in BCIs, but in technology overall. This may not have been that clear in my presentation, but in the multimodal condition, the vibrations elicited something called motor illusions. What you can do when you vibrate at a certain frequency is make the illusion to the participant that their hand is closing. So in our sense, this would be the most transparent with the motor imagery component for the participant. Obviously, it was not the case here, but this was the goal that we had when we designed it.
So as a follow-up on this question, can we perhaps assume that the multimodal feedback was not as transparent as you expected it to be?
This is one interpretation that we went for with the paper. I think, yes, the mismatch between the 2 components, the temporal mismatch, we initially thought that it would be better, so that they would sum up towards something more informative for the participant. But ultimately, they were distracting, as you said this morning. Sometimes the interactions between the 2 sensory components can be quite difficult to follow. So I think in the end, yes, the transparency was not there.
Thank you for that clarification. The second question is regarding the analysis you did with the theta band and how that related to the performance on the task that healthy participants did. We're still on study 1.
Okay.
So you refer to neurophysiological specificity of beta down regulation. You also looked into the theta band, but we all know that motor imagery task as well as observation of the feedback is related to mu band activity as well. And that came up in study 2.
Yes.
I was wondering if you actually explored that in the study 1 with the data you collected from healthy participants.
So we did explore that. I have this here. You can see the alpha mu predictor on the left. So there's a key difference between the first and the second study. In the first study, we trained the participants on the 8 to 30 hertz band. So this mu activity was actually encapsulated during training, as this frequency band is quite usually done in the BCI field. But we did find this result that this drives the performance as well. But you can see this was significant in a few key frequencies. But I would say the 10 to 12 hertz component of the mu. But yes, there was a drive of the performance also from the mu band.
If I understand correctly, your feedback that was produced by the neurofeedback system was based on the desynchronization in the entire mu plus beta band.
Exactly.
Okay. That's probably why that you could not really pinpoint. Because it looks at the collective
this was part
desynchronization of the things. Okay, thank you so much. I would like to stop it here at the study 1 and pass it to my co-reviewer, and then I will come back with the following questions.
Thank you very much. So now, Professor Devos, if you have 2 questions for Claire.
Yes, I have some. Thank you very much for inviting me, and I apologize not being with you for many reasons regarding my overbooked agenda. First of all, thank you, Claire, for your invitation because of our past. And so I like it very much. As I said to the member of the jury, I am more now a pharmacologist, I'm a clinician than an electrophysiologist, but I did my PhD and I loved it. I think I was the first in the world to record directly from the electrode, the subthalamic nucleus in 1999. I published in Brain the mu rhythm, which was really amazing, and the beta was not so great. But then after Peter Brown, which is a very good man, but he kept publishing all the time beta, so that everybody now say, well, it's beta or nothing, which is stupid, and your work is showing that. So we need to be away from dogma or just that band or that band, because we know that each people, and you mention it cleverly, each people have his own, peak in different aspects and different regions. So if we keep on, the 13 to 25 hertz on beta band, we will, smash everything, and it's not interesting. But before going to the heart of my second question, I would like to remind you as a clinician, you can't say it's a dopamine replacement on your first slide, because no way, you don't replace dopamine. You are in the treatment of people. I say that for the audience, it's levodopa. So it's another treatment. It's not dopamine, and we are now trying to demonstrate that levodopa is not metabolized into dopamine. So it's nothing but dopamine replacement we are proposing to patients, dopaminergic agonist as well. And DBS is only a very, very, very small part of the population who are treated. So if you said, well, in PD, we have dopamine replacement and DBS, as a clinician, said, no, not at all. It's not dopamine, and DBS, it's marginal. But it's very interesting tool for your subject. And I like it very much because neurofeedback is really interesting. And so my first comment is that now I would love to look at and to go back to, first of all, it was clever to trying to find the explanation for the high failure rates. And I like it very much the way you move forward and try to improve the neurofeedback. And at the end, finally, the conclusion is, well, there's there's one neurofeedback for one patient, in fact, at the end. So you are to become a tailor, because for each patient, you will have to tailor each feedback, right? On the frequency, on the system, as you said, with a choice, etc. So, and it makes sense because it's cognitive neurofeedback. Now we are designing a neurofeedback just for one patient, according to that. My first question would be, could you mention the different bias in PD you may face to explain the different results you have?
Just thinking of the top of my mind, I would say the biggest bias would be placebo effect.
Right.
I think I'm familiar with the fact that sometimes you could have a high placebo effect even from levodopa for the patients. And this is very famous as of an effect in neurofeedback as well. So you have the new technology, you have all the researchers very excited around you. So you have to implement a control condition in the future that is called sham feedback. So you give, in a way, fake feedback and you test whether this is the closed loop that improves the patient or just the new technological aspect and the excitation of the patients.
Yeah, you are totally right, Claire. I think I mentioned that, as I'm doing a lot of clinical trials, you can't, say anything. And the rate of placebo effect is 1/3, is 33%. So it's magical, in fact. And with the white coat effect also we are doing that usually daily. And also with neurosurgery, you can even reach 50% of placebo effect. So for instance, the grafted cells, we know that the grafted cell is not working at all, but the patient are really, they love that and they feel that they are better. There are also a couple of other bias. The DBS position, Brian Lowe is in the room, and we know that the neurosurgeon knows that very well. Maybe you should have the active contact and with the Yeb Atlas, you may position the contact active because it may change on DBS what you are doing
So just
when you're recording.
We didn't give the feedback on the deep brain stimulation electrode.
I know, but you are recording from the electrodes, right? No?
From the cortical electrodes. So we have important effects to look into with the precise localization. Even with EEG, there's a lot of spatial smearing. But
not for DBS. Okay. The dose of levodopa you are doing, because we know it's an inverted U curve, and you have motor effect. But if you want to have higher motor effect, you can. It is detrimental for attention. So in your case of neurofeedback, they take the levodopa is increasing and then down, and so you have to check in the future when you assess the patient according to the dose of levodopa and the time after the dose of levodopa, because they are
a really
the
important
same.
question. We did it. We recorded the time of the at which time the patients took their treatments, and we also have the dopaminergic dosages. So the dose did not predict the performance, but the responsiveness of the patients to their dopaminergic treatments did. So the patients which had the best dopaminergic responses for their motor symptoms were the ones whose performance was the best.
Okay. So, but also the time after the dose, if it's 1 hour after the dose, it's the best moment to assess them.
So this is what
because there's circadian effects, and you mentioned age effect. Now, I want to finish on this. This probably the most important point for me, and we can discuss the design, the imaging, etc. How we can improve that in the next question. But you mentioned at the end, well, we have no correlation with agency for PD. And now I take the hat of the physiopathologist of PD. Do you know what is a PD? In fact, if you want to summarize what is Parkinson's disease. Parkinson's disease is a disease of automaticity. This is Parkinson's disease, automaticity. So indeed, as you mentioned, there is an abnormal sensory motor integration, that's for sure. But if you do that, and we are doing that for the patient every day. If you do it 7 times or 10 times or 20 times, you won't have the same result. Because the more you do, the longest time do that, the more you are in an automatic way. And you're failing more and more if you keep on doing the same movement. So this is really important, and that exactly explain the key result you have at the end of in PD people. Okay, I don't want to be too long, otherwise Raphaëlle will shut me down. So I let the time for other people, and we will come back to you after.
Thank you.
Thank you very much, David. So now we can have questions from Dr. Rimbert.
Yes, so thank you, Claire, for the invitation. So it's my first time as a jury member. So I have a lot of questions, and especially about ERD and DRS. You show in your presentation and also in your manuscript. So do you think that the ERD you showed for neurofeedback condition and motor imagery condition, so especially for neurofeedback condition, do you think that the ERD generated could be due to the neurofeedback and not for motor imagery task? Because in the second study, you showed result with very big ERD in the beta and mu rhythm for neurofeedback condition, but not for motor imagery task.
Yes, you can see. So there is still desynchronization in the beta band on the left motor cortex in the patients with only the motor imagery aspect, but it's much less strong, significantly less strong. But in the mu band, we did not find this effect. I think one thing that should be mentioned is that the patients alternated between these trials. So maybe we should look into whether the patients could have preferred the neurofeedback and whether they would disengage during the motor imagery only compared to just taking a patient and asking them to do the motor imagery. Maybe they could disengage due to the comparison.
Okay, thank you. And did you look about variability between user from the first study, but also for patient in the second study, in terms of localization, but also in terms of frequency band, where the ERD is much larger?
So I have yet to look precisely at the really deep and specific neurophysiological modulations. These on the right are individual data points. So you can see even in the patients, there's quite a lot of variability. You have a lot of reduction that are best during neurofeedback, but you also have synchronization in the beta band as well as in the mu band. There's quite a lot of patients that did not have a lot of desynchronization. So showing these topographical maps individually for each patient and finding a way to put them together and analyze it, I think is an important step. I have not yet performed, but I would be very interested to see.
Okay, thank you. And final question I have is about results you show in your manuscript for the neurofeedback and vibrotical stimulation. And you show that you have a very continuous ERD in the mu and beta band. And I was curious that you have no ERS in your temporal window, because sometimes when you perform motor imagery continuously during a long time, maybe sometimes you have ERD and after that an ERS.
Yes.
And in all your experiment, in the 20 seconds of trial, you have always a very big ERD.
I was surprised a little bit when I read your PhD thesis that we did not have this result. In the multimodal condition, when we looked at the precise modulation during the 25 seconds, we found that there seemed to be non-stationary effect with actually increased desynchronization during the vibrations. So this engaged the motor system and increased the ERD. What did I want to say next? Give me 2 seconds. Can you repeat the question?
And sometimes just to complete, when you stimulate with median nerve stimulation, so it's what we have done in Bordeaux, but also with vibrotactile, sometimes you generate an ERD, and after the ERD there is an ERS. So it's why I was a little bit curious why in your temporal filter you have no ERS at all.
Okay, so I have it. What I wanted to ask you was whether in your experiment you had action observation simultaneously. So maybe I would say that the continuous action observation during the motor imagery maybe would help sustain the desynchronization throughout.
Yeah, okay, thank you.
Thank you, Sébastien. So now we can have questions from Dr. Vernet.
Hi, thank you very much, Claire, for the invitation, and thank you to your supervisors. I enjoy reading your thesis a lot. I enjoy your presentation. I really appreciate how you bridge a sense of agency world with neurofeedback. It was very, very interesting. I enjoy the care you took when you're retracing the history of how things were discovered while not losing track of the current knowledge. I enjoy how complexity and contradictory findings of the literature are carefully described while still remaining simple to read. And so great work. Congratulations.
Means a lot. Thank you.
I have tons of questions. Sometime I will play the devil's advocate, but that's not critiques of your work, of your design, of the interpretation, which are very clever, just to have your point of view.
Do your worst.
Okay. So I'll start with question on agency. I was a bit surprised that in your manuscript you don't talk a lot about deficits of agency in patients. You don't have a part on that. It comes here and there. And I would like your view on, so you talked a little bit also during your presentation. So there is a decrease of agency, which is related to the motor impairment, the comparator model being broken somehow. Yes. There is also excessive agency sometimes due to the L-DOPA treatment. So there are some kind of discrete regulation. And my first question concerns the lack of effect of agency you have in study 2, where there is no link with agency. And I was surprised by your conclusion. So you look at the global level of agency and you say, oh, we have similar levels, so agency is not impaired. And to me, the fact that there is so we know that performance is a big influencer of agency. In particular, when the comparator model is not working appropriately, we have to rely on other source of information. So we rely on performance, and here they don't rely on performance. Yes. So for me, there is an impairment of agency.
Okay.
I would like to know what you think about that.
It's impaired because you do not find the link between the two? Okay. Right. I did interpret the non-significant difference as a non-disturbance of agency. If we define agency as the relationship between between the actual construct of the performance during the task and the subjective report, you're right, there's no level of agency. I think the idea behind this comparison would stem from the classical results in the literature. I think in most of the literature, for instance, when they find this dopaminergic effect, they compare the level of agency of the patients with dopamine compared to with levodopa, compared to the resting state with their treatment. So this is quite a simple analysis in that sense, but you're right, maybe the disconnect between the actual performance and the sets of agency could indicate that there's factually a disturbance.
I have also a question that I in my mind reading your entire thesis. So you have this link that feedback transparency would lead to better agency, would lead to better performance. You question that the link, you cannot really assess the directionality of the link. Could it be that, how can you discard the fact that healthy participants are basing their sense of agency answer just on their performance. Could it be also that it's a related question, but to me, there was a condition that was lacking is doing the same task with a visual feedback that is supporting the motor imagery. It's helping them to see maybe her hand being open and closed.
But no closed loop.
No closed loop. A little bit like you have in your training at the beginning.
Yes. Yes.
Have you looked at the, I guess you don't have the agency question during the training,
No.
but you can have the performance during the training. Did you look at
Yes. For your first question, whether agency could stem from the actual performance that they experience, we had a complementary question. So we had, did you feel like you were the one in control? But we also asked them, did you feel like you were succeeding at the task? And we found that this sense of success was not the one that predicted the performance, but it was rather the sense of agency. And we also had this question in patients, and similarly, there was no link with agency or with success.
That was clever to have these two questions.
We had the same question from a reviewer, so we were really happy to have this in the questions. It saved us. And about your question about action observation with motor imagery, this is a really important factor to check. We do have the neurophysiological modulations data. It's a bit trickier to interpret because we only have 1 or 2 trials compared to the 8 to 10, depending on the experiment. So there's still a bit more work to do to reach this level. But this would answer your question, Sébastien, as well, whether this is linked to the action observation, only action observation with motor imagery or action observation, motor imagery, and neurofeedback.
Yes, that could be a big confounding, like it could
Absolutely.
be that you just have a visual support helping
Absolutely.
you.
But there's the lack of performance in the multimodal condition where they had the action observation as well. So we managed to break this effect.
Okay, thank you very much.
Thank you.
Thank you. So I'll now ask a few questions myself. Thank you very much for the presentation and the manuscript, which were both very clear and comprehensive, so that was really nice. I have a question regarding mental workload or cognitive effort. So you mentioned that maybe it could explain some theta activity in one of your studies. I wonder if you could think of an experiment that could be run to evaluate this mental workload impact in the case of neurofeedback. Do you have ideas of additional measures that you could take?
For the experiment, I would think of, something that was done in Bordeaux, where they had distractors during their neurofeedback task. So they were testing whether when you add a distractor during the task, you could have an effect of something. But this was a really clever design. I think we could quite relatively easily implement. So having the feedback on the screen and having things popping during the trial that would disrupt the concentration of the participants. Another important thing could be getting them tactile feedback, as this seemed to be very hard to follow. So this could disrupt the focus and give a lot more mental effort in the participants.
Okay, but this would mean changing the task. I was more thinking of measures that you could record from the participant to evaluate this mental workload. So
beyond the experimental aspects, I would have to look more into the workload measures. I know that you know a lot about it due to the passive BCIs. What I would do first would be reading comprehensively about this. Beyond just theta, I would say alpha drops when your attention is focused on the task. Maybe I would say frontal alpha that I would look into first additionally. We kind of looked into it, but did not interpret it.
You could also very classically just use also some questionnaires and also some peripheral measurements of cardiac activity, for instance, or electrodermal activity, something not necessarily very complex, but could be informative with respect to the stress or engagement level of the participant.
We did ask the participants whether they felt like the task was difficult, whether they could easily understand the feedback, these kinds of things. And this showed that this ease in the task was higher with the virtual hand than the other two feedbacks. So subjectively, there was also this result that was converging.
Yeah, that's interesting. Okay, and my second question would be related to fatigue, like also the effect of the number of sessions and the duration of the task and so on. So I wonder if you could re-explain briefly the duration of the task for your two studies, and if you think that the duration and the number of sessions could have impacted the status of the users or the patients.
Absolutely. So we did only one neurofeedback session for each of the two studies. And the study with the healthy participants was very long. So it was 3 hours and a half. And through every single experiment, the participants came up very rested and happy and left completely depleted. We did look into a little bit of the fatigue effect when we tested whether performance was decreasing with the order of the feedbacks. So whether, regardless of the virtual hand or the multimodal condition, whether performance was best during the first feedback, then the second, and then the third. But there was no effect of time. So I would interpret it as fatigue here. With the patients, we were more wary of this effect of fatigue. So this is why we have an imbalanced number of trials. We would ask the patients whether they felt like they could go on for a little bit more. The total duration of the experiment was about 1 hour and a half, including the setup of the EEG. So they worked for like 30 to 35 minutes.
Yeah, it could be interesting for another experiment to evaluate the impact of this duration and maybe to add more sessions, because we all know that the first experiment we run for any kind of experiment, I think, with humans, as you said, they could be excited, there could be some placebo effect or a novelty effect. In robotics, we say also a novelty effect or wow effect. I don't know how you say that in English, but yes, so having multiple sessions could be interesting as well. Okay, thank you very much. We will start the second round of questions, if that's okay. And if you're not too tired, Claire, maybe we can have 2 questions per member again. So we'll start again with Maryam.
Yes, okay, thanks for the second round. So I would like to pose my question related to study 2. But before that, I would like to ask a clarifying question about your experimental design so that I can formulate my question better. And this is a question that will revisit the agency topic that Maureen touched upon earlier. So when I look at your procedure timeline in figure 1 of this chapter, and the design, also the text, what I see here is that you mentioned after each run of 1 motor imagery, and so this is motor imagery only, and 2 neurofeedback trials, followed by the motor task. Patients rated orally their sense of agency, and then the other questions. And this is while with healthy participants, you did 2 runs of 5 trials. And from my own experiments with body ownership, illusion, and agency, we know that repetition of this task is required for the sense of agency and ownership to develop, right? So I would like to target this point that maybe you really did not induce agency in the first place. And maybe that's yet another explanation while you don't see the same relationship between agency and performance that you evidence with healthy participants. Do you have a clarification on this? Or maybe can you perhaps discuss your own view of your experimental design? Why is it that you decided to only do 1 motor imagery followed by 2 neurofeedback trials and not more?
So we repeated this sequence, right, for 3 to 5 times. And so we asked the question for agency 3 to 5 times as well. I'd like you to clarify. So you were saying that based on
let me formulate my question correctly. Do you think that you gave enough number of attempts to your patients to fill the sense of agency that is required after. And on top of that, motor imagery only trial, you didn't even give any feedback. So that is like 1 motor imagery, 2 neurofeedback, and then you're asking them to clarify sense of agency. Do you think that you really measured sense of agency in this experiment?
So I try to be as clear as possible in the question that I was asking the patients, really trying to hammer the meaning of the question and probe them sometimes with their response. There's one thing in the patients that we noticed is that they have, most of them, more difficulty than healthy participants to rate for any scale. They have more difficulty to set a specific number. So that may also be one interpretation, that their subjective reports could be disconnected because the construct of agency was not that clear, even if I didn't use that word for them. I would be interested in looking into the temporal evolution of agency, maybe looking at whether along the trials, sense of agency improved or stagnated or actually decreased based on the performance. Maybe this could explain part of non-significant results. Maybe there was this relationship at first and it degraded with the fatigue, for instance, or the opposite. For the healthy participants, they did in total 40 trials of 24 seconds. So to my understanding, they had quite a lot of time to get acquainted with the task due to their fatigue levels as well. They had quite a lot of time in it. I would trust their ratings, and based on their levels above 5, I would say most of them had this sense of agency that we could trust it.
Yeah, so let me try to push back a little bit here. I don't see any problem with the experiment design you did with the healthy patients. I think they were given enough time to habituate with the task, and the sense of agency they report should be legit. What I'm asking is that, why did you design your experiment this way for the patients, that motor imagery and neurofeedback trials are mixed and not separated in 2 completely different conditions? And if you did it already and you're not sure about the explanation, if you go back in time, how would you revisit this experimentation?
I would defend this choice of the alternance. We wanted to avoid having a block effect where they would only do one task and then switch to the other one. And we kind of constructed it in a way that switching between the 2 could abolish this sequence and fatigue effect, as well as the dopaminergic effect that David was talking about, because they take their levodopa before, so their motor state varies along the trial. So if you have all the motor imagery at the beginning, you could expect to have a strong effect of dopamine. I agree that there was not a lot of trials, so this is why it's more a feasibility than an actual deep investigation into the sense of agency of the patients with neurofeedback. But for the alternance, that's mainly why. And we also always had this issue of what control question can we ask during the control condition. We did not have a passive, I did not think of a way to ask this passive construct to find it for the other one. So this is something I'm still struggling with figuring out.
I think I'm convinced with your justification of the experimental design. I do not have much experience with Parkinson's disease, and I didn't know really the design needs to be adapted to the medication taking of the patient. So that I understand, but perhaps you can take this point into consideration when you improve your thesis, because I actually think that the agency here, there is a limitation with the measurement of agency construct that could explain why you didn't find a relationship as you did earlier. So on one hand, it's true that sense of agency is probably broken in these patients. And on the other hand, maybe it was not reliably measured in this case.
So to your understanding, the issue would be rather the alternance or the low number of trials?
The low number of trials that doesn't allow really for you to feel that agency feeling that this is a movement that I have caused. First of all, with the motor imagery trial, you didn't even show the feedback.
Yes.
There is no feedback there, right? And then that is followed by two neurofeedback trials. And normally you need to have repetition of motor, of visual feedback with the motor intentions to establish this sense of, okay, I am the initiator of the action here.
Yes.
Which you had in the case of healthy participants where they were given 40 trials.
Yes.
For different.
I would just add like anecdotal experience. We were talking throughout with the patients, and for the patients with really high levels of performance, they were reporting that they really felt embodied and they felt like the virtual hand was an extension of their own body. So I think quite a few of them managed to gain a sense of control and a sense of extension of their body into the feedback. Not all of them, but anecdotally, some of them reported this orally.
Thank you for your answer. Now I would like to move to my next question, which is more about your discussion chapter and future research recommendation. I thoroughly enjoyed some of the impactful suggestions that you presented on the discussion slides, especially how you can design these tailored neurofeedback sessions for Parkinson's disease patients. This is more of a personal question to you. How are you going to proceed with these ideas? Are you planning to already implement some of these ideas? If you are, which of these ideas and research questions are more interest and priority to you?
So I'd love to implement these ideas. A lot of patients ask if they could do another session after their deep brain stimulation operation. We would need to have the ethical approval for this second session. The first thing I would implement is the spectral parameterization I was telling you about. I think tailoring the, is it this way? Yes, tailoring the actual neural activity that we train is key, because in some patients, you have this curve that is very typical of an absence of a periodic activity. So the green and the black lines seem like background noise. You don't have any rhythmic activity, as you can illustrate when you see a peak. So adapting the frequency of the peak as well as the electrode that we find the peak in. Maybe this patient 8 could have a peak in a nearby electrode, but not on the precise C3 electrode. So this is something that would be very important. I think adapting the motor imagery task as well. So we have this kind of a one size fits all, just imagine clenching your hand, and we don't have a goal directed motor imagery. And beyond just providing choice, providing like movement that is targeted as something that is a bit more ecological than just the air, could be a good target for the future.
So do you already have a plan, like a postdoc position where you're going to implement these ideas?
I'd love to. This would be perfect. I'm continuing on the team of Parkinson's disease patients, but rather on the freezing of gait aspect. So the next step I wanted to do was go towards invasive recordings. So I'm going still in the closed loop field, but trying to do cortical stimulation, closed loop cortical stimulation to reduce freezing of gaits. So exciting opportunity.
Yes, it is. Thank you.
Thank you.
Thank you. So now 2 questions from Professor Devos.
Yeah, thank you very much. I have to admit that I agree with Maryam with the first point you discussed in the fact on the number of repetitions that can influence, as I said. If we go back to some really trivial observations that we all know when you are facing a patient as a scientist or a neurologist, if the patient is looking at you doing the same thing, you know, like the tapping movement, it largely improves the tapping. And also, as I said, if he's looking at his finger performing the trial, it also largely improves. It is also known, you will study freezing of gait, which is something relatively magic in PD, and a lot of people love that. This is the same problem. When there They have, let's say, to a very simple way, you shift from automatic motor program to a multimodal input program. And so you go with the SMA, you are really on an automatic program, which failed in PD. And so then you have to move when the disease advance, or if you're off, you have to move to a multimodal sensory input to improve. This is why, you know, you will see classical video of people with freezing of gait, and when you're doing music or, you know, acoustic impulse, then you improve the gait because it's following the rhythm. And it's the same for tapping. So we really need to have this in mind and to control the number of experimentations and exactly how he's doing it. And for sure, and you proved it with your experimentation, with a visual cue, you largely improve the system. It is really well known, and you show that. Now, how we can further improve it, and this is the key point. And what do you feel about vibration? How you interpret it? You know, why this is not good?
So
simple way, you know, simple way. Don't go into
the most simple way is just the mismatch between the visual
exactly.
and tactile that is terrible.
Exactly.
But we chose not to do it in the patients because we didn't really have time, and we had this quite discouraging effect with the healthy controls. But when I wrote the introduction of my thesis, I wondered whether this component could actually be beneficial for the patients. So Maryam talked about this morning, that the multimodal aspect can actually be beneficial to the patients. And I think someone trying to implement neurofeedback in patients might want to look at whether it's vibrations or functional electrical stimulations of the limbs. I think this addition of a sense for motor rehabilitation is really important.
You're totally right. It has to be in early PD people. Because if you are more advanced, then you worsen the situation by having multimodal activity. You will see in the freezing of gait, if it's early freezing of gait, you improve with acoustical rhythm, etc. But if you're late, you worsen the situation because now there are dementia coming, severe cognitive disorder. And this is the second point. You said providing choice of the feedback is really important. The color, the system, etc., which make a lot of sense. In fact, you know, you are tailoring a neurofeedback specific for each patient, which is probably what we need to do in the future. You're also gamifying, you said. And this is something really huge in PD because, you know, PD is not just rigidity, bradykinesia, and tremor or gait disorder. PD is it's a psychiatric disease, and in which you can have addiction or apathy. And a lot of people have, you know, lack of motivation. More important, it's also a cognitive disorder from the start. And you know what is, this is my question because I have to ask question. What is the most preeminent cognitive disorder you can find from the start of the disease?
Good question.
Very simple. It's not a trap.
Most prominent
well,
impairments in the patients?
Like you can have also with age, in fact.
Memory issues?
Before that, attention deficit.
Okay.
So, you know, you ask them to concentrate on a task and they have attention deficit. So this can also be, you know, can impede your system. So when we are doing such a assessment, we need to check all the cognitive function and all the behavioral psychiatric function to be sure that you're not enrolling a patient having some apathy or some severe attention disorders. Because at the start of the disease, some people have nothing. They are just normal cognitively. Some are just slow, but normal. And some have already, for the first years of disease, some attention disorder. So probably in your 15 people, you have some having that. And
so it's quite homogeneous population because these are deep brain stimulation candidates.
Yeah,
we have the MoCA scores, and these range from like 26 to 29. So
yeah, but you know, I know them very well because the DBS population has already advanced population.
Yes.
And they already have some apathy, slight, some, and they all have attention disorders at that stage because they are, well, my colleagues in Paris try to operate them as soon as possible, but there are still, 7 years of disease progression. So this also can explain this. And now if we go also, so also keep in mind that we need to stay away from just mu, beta, low beta, high beta. We need to have a global view of the different rhythm and then to extract from a patient this spectrum of activities that we need to have a neurofeedback on. That's
Yes.
really important.
Mm.
And finally, and this will be my very last question, even if there's another round, I will not ask any more questions. Well, no, I will stop there because I think we are done and everything was well answered. So I would
if you want, David, you can have a last one. There will be no other round, as you want.
Yeah, no. I mean, I already said what I wanted to share according to the multimodal, the neuropsychological assessment, and the what we can say, on the spectrum of the rhythms that need to be, have a global view. And I think this could change really the result and really improve them because if we consider the disorder of automaticity, obviously the problem, the agency, as Maryam said, is also, and you said that also, is broken because you think you can do it and you're not doing well. I give you an example, very simple one. When a patient is talking or walking after several years of disease progression, like before DBS or something, when his wife or her husband, they are not paying too much attention and the disorder is severe. And when somebody else comes, then they improve themselves because there's a new cue and there's something new arriving. So you see the automaticity, how it can really trigger. And if you ask him or her, are you performing well? He said, yes. But I said, no, look at you, listen your voice, it's too low. Or look at your face, you don't express yourself. Or look at your gait, your shuffle. So it does not have from the start a good, assessment of his own performance because automaticity is broken. So for sure, the debate on agency, I mean, makes sense that you find these disorders, and you can't improve that just with new feedback. I mean, agency, not the performance, for sure, hopefully. So thank you very much, Claire, and I'd like to really congratulate on the work you have done and the way you are thinking and presenting. The presentation was really nice and really, really good. So I'm happy that you are continuing in the field of Parkinson's disease with the team of Brian and with everyone. It is really great. So all my best wishes. Thank you.
Thank you very much. So now, last question from Dr. Rimbert.
Yes. So if I remember well, in your manuscript, you use a different frequency band for the neurofeedback performances in study 1 and 2.
Yes.
And can you explain me why did you choose a larger band in the first study and not in the second?
So the true answer is that when I started with neurofeedback, I had no real experience. So I used the protocol, the classical GRAT protocol, just changing the feedback. So this large mu and beta band is the reference in the field. And we did not know at this point if we were going to manage to reach the state where we have patients trained with neurofeedback. And so we did this first step. We had the pleasure to manage to get to the patients in my 4th year of PhD. And this is why for the patients, to my understanding, most of the literature focuses on this beta activity, as David pointed out. So we went with this beta-specific modulation, but you saw that humans don't care about the design. So sometimes mu is modulated as well. So even if we change the band, that didn't change the modulations.
Yeah, okay. So yeah, it's true that it was very interesting at the end of your presentation. You show that you have some differences between low beta and high beta, and also that you have a very big ERD in mu rhythm. So my question is more, do you think that it's possible to design a BCI or neurofeedback system more adaptive to the user? And is it very useful to consider only the beta, for example, low beta, or you think that mu could be used or other rhythm? What is your opinion about that?
So this is a tricky question I first dealt with during my first internship in 2017 with Nathalie. Initially, we were planning not on just doing classical neurofeedback with the threshold, but we planned on implementing classifiers. So the classical machine learning algorithm that learns on the individual pattern of the participant. And we came up with the conclusion that what we wanted was not just adapt to what the users normally modulate. We wanted to force, in a way, them to modulate the beta activity that we were interested in to obtain the motor modulations. So this was really optimistic at the time, but we cannot really force modulations beyond, to my understanding, the actual individual spectrum of the patients and the participants either. What was the question?
And do you think that maybe we can use other machine learning algorithm to try to adapt the system to the user in terms of frequency band, but also in terms of other parameters?
I think an interesting, it's quite old now, but there's something, I think from Michael Tengerman, he actually identified the neurophysiological signature during a motor task. So this allows you to individualize the neurophysiological target, but not take any neurophysiological modulation. You have directly the individual signature of the motor execution pattern. So I think going towards this, whether during motor execution or maybe during a symptom to assess the symptom-specific signature could be a big step to take.
Okay, thank you. And my last question is a more hypothetical question. If we play a game, imagine I give you a lot of money and a lot of PhD students, very brilliant like you, which study you will perform very at the beginning to continue this project? And do you think that there is a future in terms of neurofeedback system or BCI in this application of neurofeedback for patients?
I would say if I had a lot of money, the first thing I would do is trying to get the brain-computer interfaces out of the lab and into the home of the patients. So going towards more portable systems. There's a recent study that did this with mu downregulation, and they found that this seems feasible for the patients. As Maryam said, just having to come to the lab every week for several weeks of training, to me, is a major factor of exclusion for maybe patients that do not have caregivers that can take them to the lab. And it's time consuming. It's a lot of bad things for the patients. So trying to take this from the lab to the home of the patients, I think that's the first factor I would try.
Thank you.
Thank you.
There's not a lot of money.
I haven't PayPal.
Sorry, I don't see what's happening in the room. So, thank you, Sébastien. So now we can have the last question from Dr. Vernet.
Sorry. I have a question about the final target of the feedback. Because in PD patient, you don't have only problem of desynchronization of beta, but you have also a baseline beta that is too high.
Yes.
why do you target the desynchronization during movement or imagery and not the baseline beta? And an associated question is, when you target the desynchronization, does it have an effect on the baseline beta? Have you looked at that? Do you have resting data at the end of the session that could help answer this question?
Resting state data at the end of the session. Why I don't have the results yet is another limiting factor of my experience. I'm not sure how to quantify oscillations at rest, because you do not have the ability to correct easily for the 1/f curve. So this is why we do the baseline. Usually you have resting state of 3 seconds before, and you compare during neurofeedback compared to just before. So this silences the overall background level and allows you to just have the modulation. To me, it's a bit trickier at rest, but just thinking of it, we do have the power spectrums at rest. So we could just quantify maybe the amplitude of the peaks and trying to see if this peak amplitude was modulated after the training for the patients. But of course, you do have the effect of levodopa treatment going down, and we know that levodopa reduces beta. So there's a bit of a mixed bag.
Yeah, and the sham thing that you plan to implement, but so a question, it's a little bit the same question, but I couldn't figure out if the goal was to modulate beta as a neurofeedback goal or to improve motor imagery. And I was going back and forth. What are you really trying to do? And
it's a good question. It's a good question.
Do you use beta to help mental imagery, and this is your final goal, or do you use mental imagery to help beta, and this is your final goal?
Use mental imagery. If we aim to make motor imagery more efficient, I'd say we failed in the, as you can see in the modulations with this control condition. And to go back on your question, I didn't answer on this fact. What do you want to train, the ERD or just the beta? We train the beta level to reduce below the resting state, but the desynchronization is really the neurophysiological construct when you can correct. So we train beta reduction compared to resting state, and when we analyze it, we find that we have stronger ERD. So we have a bit of one spilling over into the other construct. And I missed a question.
No, that was about that. That was to figure out what was your end goal. There is a way and there is an end goal, and maybe they are not exactly aligned, and that's fine. But
I would hope that we manage to improve the motor imagery of the patients, since for the real life transfer, it's not that feasible to propose them to have real life feedback, or at least in the state of the art. So improving their ability to do just this cognitive task in real life, to me, is an important outcome.
Yes. And I have just a tiny question. Is it transparency that matters, or is it reward? And how do you disentangle both? Is it more rewarding to see?
It's a good question. To see a virtual hand?
A hand doing what you expect it to do. Maybe it's more rewarding than just a bar. So is it really the could you have something more rewarding, less transparent, but more rewarding that will work best?
This is something that was tested early on when researchers, I think in 2008, tried to improve feedback. They put smileys on the screen. They found that compared to just the bar and no smiley, it was better. So improving the reward seems to have an effect. Intuitively, I'm not sure I would consider seeing the virtual hand I implemented as rewarding. Maybe with a more immersive aspect and with actual goals. So having something more motivating, maybe score points, objectives, I would see the virtual hand environment as rewarding. In this way, I think it's a bit too plain to consider it really rewarding. But you're right, there's this factor, and it's really important with the dopamine and the levodopa in the patients. So it's something we'll have to discuss in the article.
Okay. Thank you very much,
Thank
and
you.
congrats again.
Thank you, Claire. So I have two very naive questions because I'm not an expert on Parkinson's disease, but when I read your manuscript, at one point in the beginning, you mentioned some side effects of neurofeedback in the theta band, if I'm not mistaken. So I wondered if you could tell us more about side effects of neurofeedback.
So neurofeedback overall is a quite safe procedure. There has been a lot of meta-analysis showing that beyond just fatigue or maybe headaches, there are not any side effects of the training procedure. However, during invasive neurofeedback, an interesting result from the researchers was that they found that in some patients, tremor increased during the training, and they found the correlation with the level of theta activity. So the more theta activity was that was modulated during training, the more the tremor seemed to get worse in the patients. And just below this brain-computer interface field, you see the same correlations in patients when you just measure their level of tremor and their level of theta. And I think one lead is that when you try to just assess at what frequency the patients have their tremor, this often interacts with the theta band. So you have tremor in the theta band with the patients. So I'm not really sure how you can interpret it of whether it's a cognitive effort, the patients are more fatigued, so they have more tremor. It's difficult to see the cause, but they warned the researchers in the field of this effect.
Okay. I think Professor Devos wanted to react.
Yes, I can react because I know that by heart. In fact, as you said, you are right, Claire. It's because the cognitive effort and you feel more stressful. And when you feel more stressful, all the abnormal involuntary movement are exacerbated. So it's just because indeed you feel more stress during the task. And so the tremor is increasing. So it's an indirect correlation. It has nothing to do. We know that theta is not related to tremor.
Okay, and so that means that there might be some side effects to the neurofeedback, even if it's using a BCI, or if they are stressed enough during the experiment?
I'd say this could be the other side of the coin to the placebo effect. You could have stress related to the expectation of the experimenter, for instance. You want to perform so that the experiment succeeds, and this is a common result that we found in all the patients and the participants. When you have no movement on the screen, this is really frustrating and sometimes stressful for the participants. So it's difficult to disentangle whether this is related to the closed loop or just to the experimental factors overall. So you could check the stress level between a placebo condition and an actual neurofeedback training. That would be really interesting.
And maybe it would be interesting to have a follow-up with the patients, not like a few days later, to know if there has been so many side effects. Yeah. Okay. And again, a very, very naive question. I don't know if it's relevant here, but I read that Parkinson's disease patients can have internal tremors, internal vibrations. I wonder if it's true, if you confirm that, and if there has been any study to assess the neurophysiological correlates of these tremors.
So this is part of why we also did not include vibrations for the patients. We were trying to avoid giving them the same sensation as when they're having the tremor. Some of them did have residual tremor during the training, so their right hand was sometimes rigid or shaking during the trials. So this is something we'll have to take into account in the analysis further. I'm not sure about the internal tremor, honestly. I've not heard that much about it, so I wouldn't know.
Okay, that's okay. It's like tremors inside the abdomen or yeah, inside the digestive system or so on. So they have these, some patients apparently report having these sensations, and in other diseases as well. So I recently read about that. So I wondered if you had any information, but that's okay. That's fine. Thank you so much for your answers. Thank you. And so we finished with the jury members, and now thank you very much. Thank you, Claire, for this brilliant defense. So we are happy to unanimously grant you the title of Doctor en Neurosciences. And we also unanimously congratulate you for your excellent work.
Just a minute.
So just in a few words, we were very happy with your presentation, with excellent slides and very nice flow, very smooth and in control, and also great pedagogical skills that we noted. So that was really agreeable. And also we noted 3 major contributions in scientific domain, clinical domain, and technical domains. So you have a great range of capabilities, and we want to congratulate you for that. Yes, we're very happy with both the manuscript and the defense, and we wish you great future in research. Thank you.
Thank you, Raphaëlle.